All right. Now to what we know. Again, I've said that synaptic vesicles are initially synthesized in the cell, in the soma, the cell body, and they bud off of the trans-Golgi network, just as constitutive exocytosis would at a cellular level. This is especially true for vesicles containing peptide neurotransmitters, which have to be made in the cell body through protein translation and post-translational modifications before being packaged into vesicles. And then these new synaptic vesicles travel all the way down the axon on axonal transport, which is a relatively slow process. So on those kinds of neurotransmitter packages are very slow to regenerate at the nerve terminal. And the cell has to be very cautious about and judicious about how often it expels those kinds of neurotransmitters, because energetically that's a lot of work to bring the needed neurotransmitter to the site of release. But this also gives us plenty of places that we can go to look for example systems and of membrane fusion and fission. All right, so from protein analysis types of experiments on isolated synaptic vesicles, we have come up with many, many families of proteins that live in the synaptic vesicle and at central synapses. And there's way too many of these proteins for us to talk about all of them. I'm going to focus this particular talk on the exocytotic machinery, and in the next half of talk we're going to talk about the endocytotic process. And I'm going to just let you know that there are all these other proteins, and there's more that aren't even on this diagram, whose names are out there and all have abbreviations, but we're not going to talk. The principal proteins involved in synaptic vesicle release are called snares. And the snare is a historical name. We're not going to go into where the abbreviation comes from. But let's talk about the principal ones. There are what are called vesicular snares that live in the synaptic vesicle membrane. Key one here is synaptobrevin, which is also called VAMP, V-A-M-P. The cell biologists tend to call it VAMP. The neuroscientists tend to call it synaptobrevin. The field, unfortunately, has not settled on a single name. And then there are target snare proteins which live in the plasma membrane. And chief among these in the nervous system is syntaxin, shown here in red, which is an integral membrane protein, just like synaptobrevin is. And then there's a peripheral membrane protein whose name is SNAP25. The SNAP here stands for soluble NSF associated protein of 25 kilodalton. So it has nothing to, the S has nothing to do with synapse. It was found in a pull-down assay because it bound to something called NSF. But it turns out that SNAP25 is one of the key proteins, and it only binds temporarily to the plasma membrane. The other protein shown here on the vesicular side is probably, I don't know whether this is to scale or not, it's called synaptotagmin. Synaptotagmin is our chief calcium sensing protein in this process. But there are proteins that are analogous to synaptobrevin, syntaxin, and SNAP25 for all other vesicular fusion systems. And so especially ones for viruses which are trying to invade cells have proteins of comparable structure and sequence, and these vesicular and target proteins have their cytoplasmic portions basically twist together like twisties, like you would twist a twisty to tie off a bag. And in that process, they bring the synaptic vesicle very, very close to the plasma membrane. So close, some people say they distort them. I'm not so sure about that. Synaptotagmin has two calcium binding sites in its cytoplasmic portion, C2A and C2B, and it's thought that the calcium channels, the voltage-dependent calcium channels, which are very closely apposed to the active zone, when they open and bring calcium in, that calcium binds to synaptotagmin, which somehow or another distorts the process even further so that the fusion event occurs. Now, this particular diagram is quite old and is not accurate at this point in time. So a more accurate diagram looks like this. The synaptotagmin is not pictured here, but we still have synaptobrevin, and syntaxin is the orange, and the yellow and the green are the two halves of SNAP25 that have begun to twist against the syntaxin, and the synaptobrevin is now also beginning to twist. And it's thought that that twisting brings enough force with it to bring the vesicular membrane close to the plasma membrane, and then the conformational change occurs in the synaptobrevin and the syntaxin, so that instead of being bent molecules, they straighten out, and that causes the two membranes to fuse, and a fusion pore of a temporary lifetime is opened. Now, this isn't totally accurate, but it is at least a more contemporary picture of how we envision the process. Two of the very important tools that have helped us to understand this snare protein binding to each other, and when the proteins are twisted, they're called snare pins, is the fact that these proteins are targets of some very potent toxins, botulinum toxin and tetanus toxin, all of which have different components in the venom that creates them, will bind to different sites on these cytoplasmic tails of both syntaxin and synaptobrevin, but only when they are unbound. When these are tangled up in snare pins, then the toxins cannot cleave these various proteins. So that gives us a very good assay for when the proteins are twisted together versus conditions in which they're not bound together. All right, so let's look at some of the experiments that gives us this idea of what's going on. In this very early experiment, synaptotagmin was mutated in Drosophila muscle, and to show that the C2A and the C2B binding domains were indeed calcium sensors necessary for synaptic transmission, different mutants were made that truncated the protein in the middle of different parts of C2A and C2B. And then synaptic transmission was tested at the Drosophila neuromuscular junction electrophysiologically by recording from the muscle, the flight muscle. Some of those recordings are here on the right at different calcium concentrations. And essentially at each calcium concentration, a quantal analysis was done so that one could plot M, that mean quantal content that we calculated, against the amount of experimental calcium that was present. Now, as the calcium increases extracellularly, one gets more release, so the number of vesicles, the average number of vesicles being released, increases. And under normal conditions, that has a very steep slope. This is a log plot. And the slope there is indicative of the exponent of calcium. And we see that calcium has almost a fourth power relationship. In fact, the word on the street since when I was a graduate student is that there's a fourth power relationship between calcium concentration and transmitter release. It probably these days is more like 3.5 in that exponent as opposed to 4. And so here you see some of that kind of data. But when we start looking at the various mutants with neither C2A or B, or just C2A, or some parts of C2B missing, you can see that there are different slopes to this calcium or quantal content versus calcium curve, indicating the necessity for these binding regions on synaptotagmin to bind calcium, because in their absence, the slope, the calcium dependence for transmitter release, is greatly altered.